cloning genomic DNA. And here we will focus that how we can produce genomic libraries by using or by cloning DNA in bacteriophage lambda. Suppose that we are going to clone a particular gene from human genome, and human genome, it is comparatively larger in size as compared to other organism. And if we want to screen our clone of interest or a particular gene from a complex source of human genome, then we have to digest human genome into discrete fragments. For example, if we will use a restriction enzyme like EcoR1, then it will produce DNA fragments having approximately 4 kilobase fragments of DNA. And if 4 kilobase DNA fragments, they are ligated with bacteriophage lambda, then how many clones are required to generate human genomic library? So approximately 7 into 10 raised to power 5 recombinant clones are required, and that are huge in number. And to screen this human library, it is a laborious process. First, we can compare the genome of different organisms. We can start from Escherichia coli, that its genome size is 4 into 10 raised to power 3 kilobase, and mainly it represents haploid genome where applicable. And yeast is 1.35 into 10 raised to power 4, and then another model plant system, Arabidopsis, tobacco, wheat, zea mays, Drosophila, mouse, and finally human. Here it is having genome size of 2.8 into 10 raised to power 6, and approximately we require around 7 into 10 raised to power 5 DNA fragments to produce suitable human genome library that will represent the whole genomic setup of the human genome. If we will attempt above strategy to screen a particular gene from the complex human genome, then we can face different problems. The first problem is that if the gene it is of larger size, then it may contain more than one target sites of a particular restriction endonucleases like EcoR1. So in this case, our gene of interest may be obtained in 2 or more fragments that are not suitable for cloning. And sometime even the size of the gene, it is very small, like 4 kilobase pair. If we will attempt to clone it, then we will have, you can say that, a huge number of recombinant clones that are very difficult to screen. On the other hand, if the gene of interest it is very large, then sometimes it is very difficult to clone into a suitable vector. This problem can be overcome by using suitable cloning vector like the bacteriophage lambda, as I told you earlier, that it can accumulate around about 20 kilobase DNA fragment. And here we can digest the fragment or the source DNA randomly, so that we have random DNA fragments that represents the whole DNA genome of human or from any other source, and then it is ligated with bacteriophage lambda that can accumulate around 20 kilobase pair, and then it can be screened further. Now the question is that how we can generate DNA fragments of appropriate sizes. Here basically 2 strategies they are mainly used. The first one may be that mechanical shearing, where we can disintegrate DNA mechanically by using suitable devices. But here, although the random DNA fragments they are produced, but their size can be controlled, but their cloning with suitable vector, it will require some modifications like linker molecules we may have to use to clone into a suitable cloning vector. The most appropriate suitable technique is that we should use restriction endonuclease to generate discrete DNA fragments. So another strategy that was devised by Maniatis and co-worker to generate DNA library we are going to discuss here. So in this strategy that was devised by Maniatis and co-worker, the genomic DNA or the source DNA, it was digested with two restriction enzymes like He3 or ALU1. These restriction enzymes, they can generate the blunt ends. And here, if we will allow complete digestion, the DNA fragments will be produced that is having very small length like 1 kilobase. And here we have to allow partial digestion. Partial digestion means that low incubation time, so that the enzyme will get less time to cleave the target DNA, so that we will get larger DNA fragments that may be around 20 kilobase. And then this target DNA, it is treated with EcoR1 methylase, so that internal E. coli sites it will be methylated, and the E. coli further will not be able to digest it. After methylation, the target DNA that is around, now fragment is around 20 kilobase pair, it is ligated with linker molecules that contain EcoR1 site. It is digested with EcoR1, so the sticky end will be produced, and then it is ligated with bacteriophage lambda DNA that has been cut with EcoR1. So they are ligated, and then they are packaged. They are packaged into the phage head, and then they are used for infection to E. coli, that is the host organism. So this is the strategy that was devised by Maniatis and co-worker to produce representative genomic libraries. The first step is that the source DNA, that is eukaryotic origin, high molecular weight, it can be digested with two restriction enzyme, He3 and ALU1. So partial digestion is allowed, so that the DNA fragments that are generated, they should be around 20 kilobase pair that are suitable to clone with bacteriophage lambda. After fragmentation, it is treated with EcoR1 methylase. So internal EcoR1 sites, they will be methylated, and EcoR1, it is no longer digested because of the methylation. And then the linker molecules, they are joined that contain one target site for EcoR1 on right and left side. Then it is treated with EcoR1, so that sticky ends they are produced in this DNA molecule. And then on the other hand, lambda replacement vector like the Chiron 4A, it is used as a cloning vector, and it is digested with EcoR1, so that the sticky ends they are produced and internal fragments they are discarded, and then this cohesive end of the bacteriophage lambda, they are ligated with the source DNA. And in the next step, the source DNA, it is now ligated within the cos site of bacteriophage lambda, and then it is used for in vitro packaging. No phage particles are produced that can be used to infect E. coli or other suitable host organisms.